everyone and welcome back to my YouTube channel. My name is Pranati, second year cybersecurity student studying at Sri Venkateshwara College of Engineering, Bangalore. Today we are going to discuss about the analysis of variance, which is also called as ANOVA. Today's topic contents are the introduction, statistics, technique of analysis variance, classification of analysis of variance, in which the types are one-way classification, two-way classification, applications of analysis of variance and its references. Introduction. The analysis of variance, ANOVA, is developed by R.A. Fisher in 1920. If the number of the samples is more than the two of the z-test and the t-test cannot be used. The technique of variance analysis developed by the Fisher is very useful in such cases, and with its help, it is possible to study the significance of the difference of mean values of the larger number of samples at the same time. The techniques of variance analysis originated in agricultural research, where the effect of various types of fertilizers on the production had to be studied. The technique of analysis of variance was extremely useful in all the types of researches. The variance analysis studies the significance of the difference in means by analyzing the variance. The technique of analysis of variance as developed by Fisher is capable of fruitful application in variety of problems. H0 variability between the groups, and this means they both are equal as shown. What is F statistics? ANOVA measures two sources of variation in the data and compares their relative sizes. Variation between the groups. For each data value, look at the difference between its groups, mean and overall mean. Variation within the groups. For each data value, we look at the difference between the value and the mean of its group. Now let us understand the technique of analysis variance. The technique of analyzing the variance in each case of single variable and in case of two variables is similar. In both cases, a comparison is made between the variance of sample means with the residual variance. However, in the case of single variance, the total variance is divided into two parts: variance between the samples and variance within the samples. The later variance is residual variance. In the case of two variables, the total variance is divided into three parts, that is, variance due to variable number 1, variance due to variable number 2, residual variance. Moving forward, we are going to see about the classification of ANOVA. The analysis of variance is classified into two ways: one-way classification, two-way classification. One-way classification. In one-way classification, we take into account only one variable, say, the effect of the different types of fertilizers on the yield. Other factors like difference in the soil fertility or the availability of irrigation facilities, etc. are not considered. For one-way classification, we may conduct the experiment through a number of sample studies. Thus, if 4 different fertilizers are being studied, we may get 4 samples, say 10 fields each conduct the experiments. We will note down the yield on each one of the field of various samples, and then with the help of F-test try to find out if there is a significant difference in the mean yields given by the different fertilizers. Moving on to two-way classification. In one-way classification, we take into account the effect of only one variable. If there is two-way classification, the effect of two variables can be studied. The procedure of analysis in two-way classification is total both on the columns and the rows. The effect of one factor is studied through the column wise figures and the totals, and the other through the row wise figures and its totals. The variance are calculated for both columns and rows, and they are compared with the residual variance or the error. Now in this slide, we can see the ANOVA table for the two-way classification, wherein the source of the variance is classified as between the samples, that is columns, between replicants, that is rows, within the samples, that is residual, and the total. And we have the next column, that is degree of freedom, which is C-1 for the between the samples, R-1 between the replicants, and C-1 into R-1 for within the samples. N-1, which is the total of the degrees of freedom, gives the total. The next column, we have sum of squares, which is classified or given values to the each row as SSC, that is B minus D, SSR, that is C minus D, SSE, that is SST minus SSC plus SSR. By using this table, we can easily find out the F ratio. Now, let us move on to know more about the applications of ANOVA. Similar to t-test, more versatile than the t-test. ANOVA is the synthesis of several ideas. The statistical analysis depends on the design, and the discussion on ANOVA, therefore, includes common statistical designs used in the pharmaceutical research. This is particularly applicable to the experiment, otherwise difficult to implement, such as in case of the clinical trials. In the bioequivalence studies, the similarities between the samples will be analyzed with ANOVA only. Pharmacokinetic data also will be evaluated using ANOVA. Pharmacodynamics, what drugs does to the body, data also will be analyzed using with ANOVA only. The means we can analyze our drug is showing significant pharmacological action or not. Compare heights of plants, compare birth weight of deer in different geographical regions, compare responses of patients to the real medication versus placebo, compare attention spans of undergraduate students in different programs at PC. Now we come to the general applications where we find the industries such as pharmacy, biology, microbiology, agriculture, statistics, marketing, business research, finance, mechanical calculations, and many more. So that was it for today's video. Do like, share, and don't forget to subscribe.